Hello everyone, my name is Ivania Valdes. I'm a PhD candidate on integrative genomics, and today I'm going to talk to you about a little bit of my thesis work called Role of the Intra-tumor Microbiome in the Non-Small Cell Lung Cancer Immune Microenvironment through a multi-meta-omic analysis in Chilean patients. So cancer and lung cancer specifically is one of the leading causes of death worldwide and in Chile, and most of the microbiome research related to the area has been done at gut level uh, and its incidence and disease progression and has been established regarding actually the progression of the disease and even oncogenesis. However, the microbiome present in another tissues such as the lung haven't been well established in those matters, and we don't know if the microbiome is behaving the same way as in the gut microbiome. That's why I wanted to know the link between the tumor microbiome and lung cancer and how this lung microbiome could be involved in the patient immune response against the disease. For approaching this subject, we chose to work with a lung adenocarcinoma Chilean cohort patients that had their tumor samples and also, in some cases, their non-tumor adjacent tissue samples. We had two groups of samples. Some of them were fresh frozen samples, and the other ones were formalin fixed paraffin embedded. For the first group, we had tumor and non-tumor adjacent tissue, and for the second one, we only had tumor samples that were divided into their histological grade of cancer cell differentiation state into differentiated ones and undifferentiated ones. And for this first step of the project, we did a 16S RNA sequencing analysis. And only for a subset of sample patients that had paired samples, we performed a different type of omics for the same tissue. In this case, we did transcriptomics, proteomics, and metabolomics. But I'm going to show you all the results regarding 16S RNA sequencing, proteomics, and metabolomics today. As for the methodology of the 16S RNA sequencing, we perform an Illumina sequence and the analysis of the FASTQ files. We did it using a cold environment for Chain2, RStudio, and LabC for the diversity taxonomy, differential abundance, among others, type of analysis for this part of the project. As for the proteomics and metabolomics, in both cases, we use a mass spectrometry analysis. And overall, we wanted to know or to approach the human proteome and metabolome, and in a second part, the bacteria proteome and the bacteria metabolome using these two techniques, and also using tools such as FragPy, Pig Studio, and Richard RStudio, Unibev, and MetaboAnalyst. As for the first part of the results that we had for the 16S RNA sequencing, I'm showing you here in this first part the alpha diversity analysis using the Shannon index, where for the first group, using the fresh frozen sample, we didn't get any differences, any evident differences. But for the second group, we had a higher alpha diversity for the group of adenocarcinoma differentiated samples. However, we wanted to know not only the diversity analysis, but if we had the presence of a specific bacterial member enriched in each group, and in each case that is going to be representative for that group. In the case of the fresh frozen samples, we had an enrichment using a LEFC analysis using LDA score, and we found that in the tumor samples, we had an enrichment of Pseudomonas, which has all been related with immune respiratory diseases and that promote a pro-inflammatory profile in that tissues. And for the same analysis using the formalin fixed paraffin embedded samples, we found interestingly a depletion of Akkermansia mucinifila in the adenocarcinoma undifferentiated samples that were present only in the lung adenocarcinoma differentiated ones. As for the analysis of the host, considering their host proteome, we did a filter for those proteins that were related only with some immune processes and immune cells. And as you can see here, after that filtering process, we got a list of proteins in this heat map that were decrease in the tumor or increase in the tumor or increase in the non-tumor adjacent tissue or decrease in the same tissue. Specifically, that caught our attention that we found decrease in the tumor at TCDA proteins related, and HLA class 1 is the compatibility proteins related with that process. And for the functional analysis involved related with these proteins, we found a downregulation of processes within the tumor related to the MHC protein complex binding, which is promoting an immune evasion profile. As for the microbial derived molecule, we found some peptides and metabolites using the proteome and the metabolome that maybe promote an immune suppression profile, since at the tumor level we found some proteins related with pseudomonadota, as the same that we found as pseudomonas in the first omic that we proved this, and some metabolites that were enriching the tumors, such as adenosine and inosine related with the purity metabolism. Finally, um, we found some differences between the microbial composition in the tumor sample and the non-tumor samples, with the increase of pseudomonas in the first one, which will be potentially giving rise to a pro-inflammatory profile. And also, we found that the answer of akkermansia in the tumors may contribute to an undifferentiated cancer cell state, worsening the disease. So, in another lab, we had a reduction in the TCD8 cells and HLA class 1 related proteins in adenocarcinoma samples, maybe suggesting that the regulation pathways within the tumor related to the MHC complex binding that will contribute to tumor evasion and ultimately contributing to tumor progression. Finally, some microbial peptides associated with pseudomonadota and streptomyces are consistent with our initial findings of 16S RNA sequencing and their involvement in transmembrane transport activity, and also with an increase in purine microbiome metabolites that could foster an immunosuppressive microenvironment that could contribute to the tumor progression. The acknowledgements for this project, both universities that I'm working on, and the MD Anderson Cancer Center at the USA. So thanks for listening.